So this unit is going to talk about the structure of DNA, and then we're going to go into DNA replication. We're going to talk about the chemical components of DNA, we'll talk about the scientists who solved the structure of DNA, and what they learned from the double helix model. This shows you a picture of the basic nucleotide structure. A nucleotide is a monomer, or the smallest unit of a nucleic acid. Remember that nucleic acids are one of the 4 biomolecules. So DNA is made up of nucleotides that are joined into long strands or chains by covalent bonds. You can see that the base in this case is the A for adenine. There's a deoxyribose sugar, which is a pentagon shape, and a phosphate group. There are 4 different types of nitrogenous bases, or nitrogen bases. They're adenine, guanine, cytosine, and thymine. The basic nucleic acid structure is the phosphate group, the sugar, and one of the 4 bases. So in this case, in this diagram, you can see that there are actually 4 different nucleotides. And again, the nucleotide is the smallest unit or building block of a nucleic acid, such as DNA. The black lines that you see connecting the sugar and the phosphate group, these are covalent bonds that attach the nucleotides together. Chargaff was a scientist who the rule was named after, Chargaff's rule. Chargaff's rule says that for the same number of A's, you'll have the same number of T's, and however many C's you have, you'll have the same number of G's. What he realized as he was studying the DNA structure, he saw that if there were 68% of a strand, 68% was adenine, 68% was also thymine. And if there were 24% guanine, there was also 24% cytosine. So he came up with the base pairing rules, and A will always bond or pair with T, C will always bond with G. An easy way to remember this is that apples grow in trees, A goes with T, and carrots grow in the ground, or C's go with G. So you'll always have that base pairing rule. Rosalind Franklin came along and was able to X-ray DNA, and so you can see from the picture on the bottom right, this is a cross-section looking down at a DNA strand. She saw that you had a phosphate group, well, you had the phosphate group and the sugar around the outer circle there. She saw that she was able to tell that that double helix was twisted and the nitrogen bases were near the center of the molecule. This led the way for Watson and Crick to come up with a basic DNA model that we use today. As you recall, chromosomes are your DNA in a tightly wound and condensed package. The DNA is twisted and wound around histone proteins, which we call nucleosomes. These are coiled and then coiled again in what we call supercoils to create or condense into what we call chromosomes. In this picture, you can see sister chromatids where the individual chromosomes have already duplicated during the S phase of the cell cycle. So our double helix is in what we call antiparallel strands, which means that while the left side goes one way, the right side will go the other way, and you'll see this when we pair the nucleotides together. The double helix, or the nitrogen bases, are held together by hydrogen bonds. So your guanine and cytosine, your adenine and thymine, these are all held together by hydrogen bonds, which are not true chemical bonds, but more of an attractive force. So in this case, we know that the 2 strands of DNA are held together by hydrogen bonds between the nitrogenous bases adenine and thymine, and between guanine and cytosine. So next we're going to talk about how DNA replicates.